Monoclonal antibodies by Luke and Simone. Monoclonal antibodies are
often used to fight cancers, chronic inflammation, infection, and cardiovascular diseases. Monoclonal antibodies represent a novel way to target specific mutations and defects in protein structure and expression in a wide range of diseases. A collection of identical proteins from a single type of plasma cell bind to a single epitope on a particular antigen. First produced in 1975 by KOhler and Milstein, it was a further 11 years before monoclonal antibodies were licensed for the market. Over the preceding years, 30 monoclonal antibodies have been approved for the use in clinical practice, with many more currently being tested in clinical trials. The industry has expanded exponentially over these years and is now worth approximately $20 billion per year globally. Antibodies combine to specific antigens. Once attached, they recruit other parts of the immune system to help destroy the cell. Researchers are able to design and make clones of antibodies specific to particular antigens, hence the term monoclonal antibodies. There are many different therapeutic effects to monoclonal antibodies. Typically, these mechanisms involve blocking the function of infected cells, modulating the cell's signaling pathways, or making pathogens more recognizable to the body's immune system. For example, many monoclonal antibodies are developed to make cancer cells more identifiable to the immune system. Cancer cells develop from normal, healthy human tissue and can be difficult for the immune system to spot. Some monoclonal antibodies bind directly to these tumors, making them easier for the cells of the immune system to find, target, and hopefully kill. Furthermore, monoclonal antibodies can also modulate cell signaling pathways. Cancer cells often produce a large amount of molecules called growth factor receptors. These sit on the surface of tumors, allowing them to divide. Monoclonal antibodies have been developed to inhibit the growth factor receptors. Current knowledge indicates that mAbs do not elicit direct effects on metabolic clearance pathways for small molecular therapeutics. However, the immunomodulatory properties of mAbs can indirectly alter clearance for certain small molecule entities through the attenuation of non-catabolic enzymatic pathways. Having found a healthy organism, commonly a mouse, it is injected with a relevant antigen. As the infectious agent enters the body, the immune system will produce and bind identical antibodies to fight the single epitope of the antigen. Drawing blood from the spleen, all types of plasma cells will be removed and screened before the desired cells are isolated. Plasma cells alone will die when taken out of an organism. Therefore, the healthy cells need to be mixed in a solution of polyethylene glycol and fused to tumorous myeloma cells to become immortal. Hybridoma cells are the name given to the resulting cancerous plasma that produce the monoclonal antibodies desired. The filtration process is undergone until a collection of solely the desirable mAbs are present. These can now be mass-produced. They could be injected back into the organism, they could be frozen for later use, or even modified to have additional effects. Chemical coupling of effector molecules is not uncommon. Monoclonal antibodies carry the risk of immune reactions, such as acute anaphylaxis and serum sickness. Those used in the fight against cancer see most side effects during the therapy itself. Toxic reactions such as fever, sweating, and chills are more commonly reported than skin reactions. More broadly, the side effects include allergic reactions such as hives or itching, flu-like symptoms, muscle aches and pains, nausea or vomiting, diarrhea, skin rashes, and low blood pressure. A particularly interesting aspect of antibody efficacy is its unique pharmacokinetic characteristics once inside the body. For example, it has been noted that the bioavailability of IgG in plasma is partially dependent on its interactions with the neonatal Fc barbell receptor, and such the effector functions can be improved by genetically engineering the Fc region to contain point mutations or glycan modifications. Monoclonal antibodies present an attractive option for the development of new therapies and molecular drug treatments against a wide variety of common diseases due to their speciality, flexibility, and profitability.